We developed a really strong expertise in nano manufacturing, which is basically how can I make these nanoparticles at a large enough scale to be useful in the clinic.
This is the James Cancer-Free World podcast. I'm Steve Wartenberg, and my guest is Dr. Jessica Winter. Jessica is an Ohio State Distinguished Professor of Engineering, a member of the fairly new Ohio State Cancer Engineering Center, and she is an expert in the growing field of nanotechnology. In episode 150, Matt Ringel, the co-director of the Cancer Engineering Center, he talked a little bit about nanotechnology and how it could be used in treating cancer patients. And so we thought we needed to learn more about this fascinating new field, and we invited Jessica to fill us in. Welcome to the podcast, Jessica.
Thank you. Glad to be here.
I'm really excited because nanotechnology, you hear about it, and it's something that's out there and sounds so advanced and technical and amazing, but like so many other people, I don't really know anything about it. So fill us in with the very basics. Give us like an overview on what it is, and maybe a little bit about what it can be used for, and then we'll jump into medicine and cancer.
Okay, so technical experts define nanotechnology as something that has a size between 1 and 100 nanometers. So that's the technical definition. And just for reference, a protein in your body is probably 4 or 5 nanometers. A DNA strand is 2 nanometers. A biomarker is 10 nanometers. And the fibrils that make up your tissue are more like 200 nanometers. So just to give you a range. So you can fit 5 million 5 nanometer particles in a single cell. So they're much smaller than a cell. You'd burst the cell, but...
Wait, you said you could fit 5 million?
5 million 5 nanometer particles inside.
What does that mean, 5 million 5?
5 nanometer sized particles. Oh. You can fit 5 million of those inside a cell.
Oh, so 25 million total.
No. Well, it's a 5 nanometer particle. I have to give a diameter.
Oh, okay. Oh.
Okay, so you can fit 5 million nanoparticles that are 5 nanometers in diameter inside a cell.
Oh, okay. Wow. So they're microscopic.
Right. And so in terms of their general application, I think originally there was a lot of interest in microelectronics, and that came from Richard Feynman, who's generally recognized as the father of nanotechnology. In the 60s, he gave the Room at the Bottom speech, which is really famous, that basically challenged us to think about going smaller. And a lot of people met and answered that challenge. But those types of technologies tend to be two-dimensional, flat on a chip. And I would say in the late 90s was kind of when I started my work. And we began to think about three-dimensional nano objects that we could then maybe use in new and different ways. And so that's when the first thought of using them in medicine really started to come out, was in the 90s.
So to make a three-dimensional nanotechnology device, is that the right word?
It depends what you're using it for.
This is what I'm curious about. How do you make something that small?
So most of these are manufactured using solution chemistry like you would do in chem lab. And some of the syntheses are actually quite easy. We could go step into my lab and do it in about 5 minutes, and you could see a result. But it depends what type of particle. You can make nanoparticles out of metals, semiconductors, polymers, ceramics, and they all have different uses. And so each manufacturing method may be different.
So that might be a misconception, that they're not little machines with all these parts. They're just particles. Am I close?
So you can make a nanomachine, and I'm happy to talk more about that. But I would say that technology is not very advanced right now.
And so now you're at the particle phase, creating particles. But is that the right word, or does that describe what you do?
So nanoparticle usually refers specifically to something that's spherical. You can also make nanorods. You can make triangles. You can make hexagons. You can make all kinds of different shapes.
Okay.
So we would call nanomaterial more broadly to cover like all the different types of shapes and things that you can make. And a nanomachine would be something that has a functionality. Usually we start thinking more towards nanorobotics, in that context. So something that can have input output with an external environment. It probably has some degree of triggered motion, for example. It can translocate to a different place, things like that.
How small can that be?
Well, most of the examples that people work with are still micron size, so not nanotech. As the field of nanorobotics is starting to develop, I think most people are looking at around, say, 50 nanometers. And I'm excluding microfabricated devices like in your computer chip. We can make things down to probably the 2 to 5 nanometer range using those technologies. And they can make little machines on a chip that they have like gears that turn, and I think they even made the smallest violin ever, like 10 nanometers. But those are really just curiosities. They're not very useful for
like
anything.
Proving the concept.
Yeah, exactly.
Okay, so that's a good overview, I think, and
Sure.
I'm sure everyone listening, at least I am. I'm trying to picture in my brain, in my mind, what these look like, and it's hard to picture that small, but I'm trying. So, but let's talk about the jump from, you said it started with computer chips and things like that. So let's go to the jump in medicine. Just overall, how was it applied to medicine initially, and now what are you doing now, and bring us into cancer?
So in terms of medicine, there are basically maybe three areas where it's been applied. One is imaging, and second would be biosensing, and the third would be drug delivery for treatment. And imaging particles probably emerged first in, say, the 80s. You can make iron oxide nanoparticles, and iron oxide is basically rust. That's what it is. And they can be used as an MRI contrast agent. So they've developed these where when you get the contrast agent, then they inject into you so that they can visualize cancer or other anomalies. Some of those are nanoparticles, not all of them.
So I've heard of that. I didn't think of that as nanotechnology. You're injecting something into it that makes the contrast on a screening imaging MRI look clearer, so people can diagnose things better and more accurately.
Yes.
Which is huge.
So that was the earliest innovation.
Okay.
And that's probably in the late 80s. And then in the early 90s, we had our first nanomedicine approved for cancer. It happened to be for cancer treatment. And it was basically a nanoparticle of doxorubicin, which is the bread and butter chemotherapeutic that many patients receive. And then that was quickly followed by paclitaxel, which is another bread and butter chemotherapeutic.
And I'm guessing because it's more directed and smaller, lesser side effects?
That's the vision. So it turns out there are actually a lot of problems with nanomedicine as it was originally envisioned. What I can say is you can usually increase the amount of drug that's delivered over when you don't have a nanocarrier. So if I just inject the drug, less of it ends up where it needs to be than in the nanocarrier. But there have been a number of studies that came out in the 2010s that basically showed only about 1% of the injected doses delivered, regardless of the characteristics of the nanoparticle. And it turns out, this is lemons into lemonade actually, we're really, really good at delivering to the immune system and not so great at delivering to cancer because the immune system takes up all of these nanoparticles. It views them as foreign bodies. So what did people do? They said, I'm going to make mRNA vaccines with nanoparticles because they all go to the immune system. And there's a lot of interest right now in nanomedicine for cancer immunotherapies because we're really, really good at delivering to the immune system.
Wait, I'm not sure I understand. So when you deliver the nanoparticles to the immune system, what does that do?
So you can use that to modulate the immune system as a cancer therapy. So there's a number of different cancer therapies in the immunotherapy space right now. There's a couple that nanomedicine can apply to, but one of them is we can use nanomedicine to activate your T cells so that it will attack the cancer.
Okay, that's what I was thinking. It helps the immune system better recognize the cancer cells or proteins that are cancer cells. Okay, so that's the more promising one so far.
Yes.
Okay.
So the drug delivery vehicles are out there. They're FDA approved. They get used. But the promise wasn't what we thought it would be. We weren't as efficient at delivering directly to tumors as we had hoped.
Which happens in science, and you take a step back and then take some steps forward.
That's right. The third was biosensing.
Biosensing.
Right. And so biosensor is building something usually for an in vitro diagnostic, like a little test kit. So like your COVID test that you take, that's a biosensor. And some of the earliest uses of nanoparticles in consumer products were in home test kits like that for pregnancy detection.
Oh, okay.
So the ones that can tell you 3 days sooner, that's actually gold nanoparticles. That's why they can do that. So you can build sensors to detect all kinds of things, not just pregnancy hormones. And so there are some devices where people are trying to do high throughput screening, for example, to look at maybe a gene profile or a phenotype of a patient, meaning what biomarkers are they expressing, what kind of treatment should they get based on that. So that's the other area.
Wow, could it one day detect cancer as a screening agent? You just
potentially. You may not want to talk about this, but this actually brings me to Theranos. You remember Elizabeth Holmes? So it's really funny because the professors in my department that work in this space, we're all sitting around going, it's literally impossible what she claims.
Right.
And the reason is to detect cancer, the cancer has to be there. And when I take a blood sample from you, I typically take, say, 5 mils, maybe 10 mils. The probability that a cancer cell is in there is usually 1 in a billion. And so 10 mils may not be enough blood to capture even one cell. And again, if it's not in the sample, I can't detect it. So when you say I'm going to take a pinprick and detect it doesn't matter what the technology is. If your sample does not contain the analyte, I can't detect it.
So even people who have cancer, you need a bigger sample than the drop of blood from a pinprick.
Some of the early systems that they developed, and these would improve, but they were looking at circulating tumor cells in blood, and they used patients that had metastatic cancer. So we know they have cancer, we know there's cells in the blood because they're metastasizing, and even for those patients, they had to start with 60 mils to detect the cells.
So yeah, that's something we've talked about before, and that's something that's going to advance in the next decade or more. And
so part of it is what is the limit of detection of the sensor. But the other piece is if you give me a drop of blood and there's not a cell in it, I can never detect it, no matter how good the sensor is.
Right, you can't detect.
So there's this trade-off.
Yeah, you can't detect something that's not there.
So what people are doing is trying to look at markers that have more abundancy in blood. So can we get something where we can use a smaller sample size and find what we're looking for? And those are going to be things like exosomes, DNA and RNA that's just free in blood, things like that. And so they're working on that.
Okay, great. That was a great overview. We're going to take a quick break, and then when we come back, we'll talk about what you're doing in your lab.
Awesome.
You didn't choose cancer, but you can choose where to treat it.
And when you choose the James at Ohio State,
you're picking a team of experts who understand there is no routine cancer.
You're opting for care from a highly specialized team dedicated to treating one type of cancer, yours.
A team that studies the unique makeup of your disease to develop a personalized treatment plan.
You're choosing our region's only comprehensive cancer center designated by the National Cancer Institute.
Where more than 1,700 scientists are working on new treatments and new hope for every form of cancer.
At the James, you're making the choice to have the most advanced treatments, many of which were developed right here.
And you're choosing access to the James world-class clinical trials, dedicated support services, and an unmatched survivorship program to support your life after cancer treatment.
You didn't choose cancer, but the choice of where to treat it is clear.
We're back with Jessica Winter, and we're talking about nanotechnology and cancer treatment. And I'm ready to dive in to learn about what you and your lab are doing. So I know you've got 2 or 3 big projects you've worked on. So let's start at the beginning. What was your first big project here?
Yeah, so my initial expertise was all with nanoparticles called quantum dots, which won the Nobel Prize for Chemistry this year, incidentally. And quantum dots are semiconductor nanoparticles that fluoresce. That means they light up. And we were very interested in using them as a means of detecting cancer. They have a lot of advantages over the dyes that people use now. They're brighter. And they also have a narrower color, which means if I want to detect lots of biomarkers at the same time, I can cover the whole rainbow instead of only using 2 or 3 colors. What really motivated me, I got to Ohio State, I was working on quantum dots like every other professor. I'm publishing papers, writing grants, getting money to support my work. For most faculty in engineering, that's where it stops. We publish papers and we kind of hope that maybe someone reads the paper and takes it further, but that's beyond the scope of what we do. And in 2011, I was diagnosed with breast cancer. And that was very traumatic for me.
Right.
It was ironic because I had started collaborating with breast cancer researchers about 3 months before to look at whether these particles could be used to detect circulating tumor cells in blood. And so I knew all the right people to talk to. And I had all of my treatment here at the James, which I'm very grateful for. I think I was patient 0 at the Spielman Center. It basically was opening at each wing as I needed it. So it was really great to have that experience. I mean, not to have cancer, but to have the high quality of treatment that I got here.
Wow. It's so we're glad you got to the right place at the right time and got the best possible treatment. And how many? It's 12 years?
It's 12 years now. It'll be 13 in February. So
we both knock on wood and congratulations.
But for me, that was a really defining moment in my life because I realized I can just keep publishing these papers and have a great career and it'd be fine. But I'm not really doing anything with the technologies that I'm developing. And I felt that I was uniquely gifted with the ability to fight cancer at a broader scale through my research. And I really wanted to do what's called translational research, which is where I take something from the lab to the clinic.
Right.
And that's a lot harder. There's not as much money for that grant-wise, and it's a really tough road. Developing a life science therapeutic, which is kind of the space that we're in, even though it's an in vitro diagnostic, it's not used inside the body, that's still a 10-year path, several $1 million. So facing this diagnosis, all of my friends and my researchers that I collaborate with asked me, what can we do? And I said, I want to do this. I want to do translational work. A year later, we founded a company, Core Quantum Technologies, which is still out on Kinnear Road in the Rev1 incubator. And we started working towards commercializing these particles for blood cancer detection. And it took a really, really long time. We started our first sales in January of 2020. Which obviously was problematic because of COVID.
Yeah.
The majority of customers that we would be going to, these labs that are doing cell analysis, were all hijacked to do COVID detection.
Right.
So we actually had to push out a second product, which is a magnetic version of this particle, which you can use to isolate a cell and then detect, as opposed to just detecting cells that you've collected another way. And then we finally kind of got things going after COVID kind of let up.
Well, I'm not quite sure I fully understand. Your product is used to detect cancer.
Yes.
In tumors that are removed from the body, or?
So it can be, but the primary thing that you're detecting is cancer in blood.
Okay, now in blood.
So you're taking a blood sample. You're isolating the cancer cells from it. You analyze them on an instrument called a flow cytometer, where it can show all the different colors of the quantum dots. And based on the amount of the color and the types of colors, whether it's red versus yellow, I can tell you whether you have cancer, what kind of leukemia, because there's a lot of different stratifications, and possibly use it as a prognostic tool to see if your therapy is effective. So that's kind of what the technology does. And I wish I had my pretty little picture, but I don't. But what I can tell you is it is an order of magnitude brighter than what we use now. So 10 times brighter. And that all occurs with the negative signal staying in the same place. So what does that mean? That means I can detect your cancer sooner because it's brighter, but I'm also going to have fewer false positives and negatives because there's greater space between the negative and positive signal. So this is the advantage of the technology.
So who would undergo this test? I mean, it's not for everyone yet, maybe down the road. But...
No, so this would be for someone that is suspected of having a blood cancer based on their normal blood counts. You might, or we already know they have cancer, but we don't know what type.
Okay.
So it's more of a downstream diagnostic from the original diagnostic. But you mentioned using solid tumors. You can use it on solid tumors if you homogenize the tissue. So that's another way that it can be applied.
So are we down the road? This could be a yearly test when I go to for my yearly physical. You can use what you and your partners make to see if anyone and everyone has cancer?
I think theoretically, yes, that could happen, but the biology also needs to advance. In many ways, that's where we're limited because we don't know what markers to look for.
Right, okay.
So as the biology comes along, we've got the technology and we can put them together.
God, this is fascinating. And over the next decade or two, this is what you'll be doing.
Yeah, we're really excited about this. And there are other applications for this technology. So one of the things we're really thinking about now is whether or not we can use the magnetic version for T-cell isolation for CAR T-cell therapies, for example. Because pulling out the T-cells from the blood is one of the problems that researchers face. We have a technology that pulls cells from blood. You know, it seems like a happy marriage.
Wait, so normally I do a centrifuge. You have a better way, or am I wrong?
If you want a specific cell type, you can't just centrifuge it out.
You can pull specific cell types.
Right. So I can centrifuge it out and get all the white blood cells. But if I only want the T cells, they're all mixed in. So there are other technologies people use now, mostly revolving around capturing them with an antibody. And again, that's what we do. So we're well equipped to go into that space.
Okay, well, keep going. So I know your lab is multifaceted. What's another big project?
So I still really am interested in solid tumor diagnostics because the majority of cancer patients present with solid tumors. So again, this is your breast, lung, prostate, and so on. And I collaborate really closely with pathologists here at the James, and they've communicated that where pathology is for those cancers, it uses colored dyes that are not fluorescent. Fluorescent is like your glow-in-the-dark shirt, and a colored dye is just like your blue jeans. And the reason is because the tissue from the biopsy sample has its own fluorescence, which overlaps with the quantum dots, so it's difficult to see. So we use regular dyes instead of that technology. And quantum dots are not dyes. They're not that kind of dye. So for a long time, I just didn't have a solution to that problem. But I kind of came up with something more recently, and I'm really excited about it. And we've been working on that for 3 or 4 years, and we published a paper with pathology somewhat recently showing it in cells, but still working towards tissue. So the problem is, what happens? I'm going to ask you a question. What happens in art class when you mix all the colors together?
It becomes one color.
Yeah, it becomes one color, and usually it's a dark color.
Yeah, like brown.
Yeah, brown, black. And so that's the problem. When a researcher wants to identify multiple biomarkers in a cell, say maybe I need 6 biomarkers, if I make them 6 colors of the rainbow, all the places where they overlap, I'm just going to see black or brown, and I'm not going to be able to tell them apart. So just layering colors doesn't work. And that's what we do in fluorescence. Fluorescence, we can get away with that because each quantum dot is excited by a different wavelength of light. So I can take like a red picture, a green picture, a blue picture, and put them together. I can't do that here. So what we came up with is something I'm calling erasable labeling. And what it is I label biomarker number 1, and then I erase that dye, and then I label biomarker 2 and erase that, and then I can keep doing this over and over. And by saving those images, I can compare where the markers are located in the cell.
You erase them, but the image remains in the computer.
Right, I take the image
and lay them on top of each other.
And you can layer it, and you can see the black or brown overlap, but I can pull out each individual layer and look at, where is this biomarker expressed? And that's important because personalized medicine is really advancing. So with my breast cancer diagnosis, I was treated with tamoxifen and now anastrozole. And these are molecular medicines, so this is a personalized medicine. And it's based on breast cancer patient triage of whether you have estrogen receptor, progesterone receptor, or the HER2 receptor. Each one of those is a single receptor test. I just test that one receptor.
Right.
We can do that really easily with the one dye color, no problem. But now we're starting to find medicines where I need to have these 3 together, but not this other one. So I need to label 4 things at a time to tell whether you're a candidate for that medicine. So we need better diagnostic tests to match the patient to the therapy. And that's what we're trying to do with this technology. The technology for that is pretty cool. It's a nanoparticle that's made of polymer, and we put the dye inside the polymer.
Okay.
Okay, so now we have a dye that we can use as a label.
So it doesn't seep out into the body.
Right.
The dye has a protective
really
wrapper.
Loves that polymer, so it's going to stay in that box.
Okay.
Yeah. And remember, this is all in vitro, so this is not something we inject into your body. This is all being done by a pathologist on a biopsy sample. Okay.
So you're still testing it, not in patients yet, but in the lab to eventually do it in patients.
Well, it would never be inside a patient.
Oh, okay.
It's a diagnostic.
It's always in their blood or their tumor. Okay.
Right.
Okay.
So this is for biopsy samples.
Got it. Okay.
So when they take a biopsy, it goes to the pathologist. He slices it up, and then they stain it. So that's what we're talking about.
Okay.
On top of this nanoparticle, we need a way for it to erase. So how do we do that? And what we did is we covered it with a cage built out of DNA. And so we collaborate with other researchers, notably Carlos Castro in mechanical engineering, who studies DNA as a structural material. So we're not really caring about its biological properties right now. We're using it as a building material. So we build a cage of DNA on the surface of this particle, and it has little dangling DNA strands that can bind to other DNAs. And we use that to barcode it to antibodies to bind the biomarker. And so when the antibody binds the biomarker, the particle can bind the antibody through the DNA. They hybridize to form a double helix. We take our picture, and then we add a string of DNA that has greater complement, and it kicks off the particle, and we can wash it away.
Oh, see, okay.
So the erase is based on DNA hybridization and dehybridization.
So if it's not in the patient, it's in their blood that you've removed, or in a sampling of their tumor that you removed, why do you need to erase it and get it out?
So that we can label the multiple markers and not end up with just everything black or brown.
Okay.
So that's kind of the deal.
Drug delivery is a big topic with nanotechnology, and I know that's also being researched in your lab. So what are you doing in this area?
Yeah, so when I was working on the quantum dot technologies, one of the problems that we faced was the need to make more quantum dots. You need to make enough to be useful in a patient. And we developed a really strong expertise in nanomanufacturing, which is basically how can I make these nanoparticles at a large enough scale to be useful in the clinic. And as we developed that, we started getting approached by researchers at the James asking us, hey, I really would like to study this drug that I've looked at in cell culture, but now I want to go to the next level. I want to start translating to the clinic. To do that, I need more of the drug. And I also need to be able to put it in a carrier that's safe, as opposed to the chemicals that I'm using in my cell culture now. And we specialize in that. So we've worked with a number of researchers at the James to take new therapeutics that are just being studied and formulate them for delivery to patients, so that they can be studied at a larger scale. And because we have manufacturing experience, we can make samples that are large enough for them to take the next step in testing. which they may not be able to do on their own with the methods that they're currently using.
So it's taking drugs that are already out there.
Well, they're not out there yet. They're being studied here.
Not FDA approved.
Right.
But drugs that are preclinical trial in patients.
Right.
And this is how do we deliver them safer and more accurately using nanotechnology, and you're working with them to do that. And it sounds like between the College of Cancer, the Cancer Engineering Center, the Palatani Institute of Immunotherapy, there's a lot of collaboration, and this might be a place, Ohio State could be a place where there's going to be some big advances.
Yeah, I hope so. We have 4 different projects right now, and it's amazing the different people we work with. So we do have an immunotherapy project. You mentioned immuno-oncology, so we have one in that space. But we also partner with people in public health that are facing the agriculture school studying nutraceuticals. So these are cancer preventatives. We had a whole project on black raspberry extract and whether that could prevent oral cancer. And could we formulate it so that it could be included in foods, in your toothpaste, and whatever, to help prevent oral cancers? So we span a wide range of cancer activities across this campus.
See, I think that's what I've learned today, is that nanotechnology is like a fundamental world that's going to go across so many different areas of medicine and the biology in the world around us, and you're just at the beginning, and it's going to be applied to, you're going to learn how to apply it to so many things.
Yeah.
So now you talked a little bit about your own cancer diagnosis and how, but what drives you? Because you are, your passion and your energy and your determination to come up with new things, what motivates you to just, like you said, a lot of scientists just write papers and don't take the next step, but you are taking a lot of next steps.
I don't know. I love my job. I love what I do. I love the idea that I can help other people. And I've always wanted to make real things. When I interviewed, that was one of the things I emphasized in my job interview. Like, I don't want to just sit in the lab. I want to make real things for real people. And so that's something I've always been passionate about.
And what's it like to now be making real things for real people?
It feels good. I have high standards for myself, though, so I always feel like I can do more.
Okay, well, keep doing more, and down the road, come back and tell us what you're doing.
Sure.
Thank you very much for sharing your story and your research.
Thank you for having me.
This podcast is brought to you by the Ohio State University Comprehensive Cancer Center, Arthur G. James Cancer Hospital, and Richard J. Solove Research Institute. For more information, check out our website, cancer.osu.edu.